You know, deep inside every single one of your cells, there's a secret code. And today, we're going to crack it. We're going to find out how that code gets read and turned into, well, you. This is the story of how life itself is built. It's called protein synthesis.
I mean, think about it. How can a simple four-letter code build a living, breathing thing? This is the question, right? It stumps scientists for ages. Your entire blueprint, everything you are, is written in a language with just 4 letters. So how does that ridiculously simple code manage to create everything, from the muscles that let you move to the very cells in your brain?
Well, the answer is a process that's happening right now, trillions of times a second inside your body. It is an absolutely mind-boggling scale. And that process is called protein synthesis. This is it. This is how the instruction manual for life gets read and actually put into action.
Okay, to really get a handle on this, let's use an analogy. It makes it way easier. Imagine your cell is like a giant, bustling city. And in this city, there's a super secure central library and a really, really busy kitchen. We're going to need to get to know the key players in this city.
So, first things first, why are we even talking about this? The answer is proteins. These things are the absolute workhorses of your body. Seriously. They're like tiny machines, little messengers, the scaffolding. They do pretty much every job imaginable to keep you alive. They're the finished dish that comes out of our cellular kitchen.
All right, so let's really map out this analogy. Your DNA, that's the master cookbook. It holds every single recipe your body will ever need. This cookbook is kept super safe inside the nucleus, which we're going to call the library. And the protein, the thing we're trying to make, that's the finished dish, cooked up in the cytoplasm, our kitchen.
Okay, but here's the big problem. That master cookbook, the DNA, is way too valuable to just leave the library. It's the original copy. You can't risk it. So how in the world do you get the recipe from the secure library all the way out to the busy kitchen where the chefs are waiting?
Simple, right? You make a photocopy. And that right there brings us to the first big step in this whole process. It's called transcription. This is all about making a quick, portable, and disposable copy of just one recipe.
So, transcription is basically the cell's version of a Xerox machine. The cell takes a specific section of DNA, a single gene, and rewrites it into a temporary little note. This copy has a name, messenger RNA, or just mRNA for short. And as its name suggests, it's the perfect messenger to carry that recipe from the library straight to the kitchen.
And this whole photocopying thing happens in 3 basic steps. First, an enzyme, you can think of it as our molecular photocopier, latches onto the DNA. Second, it unwinds a little piece of that famous double helix to expose the letters of the code. And finally, the enzyme just scoots along one strand, reading the code and building that new mRNA copy letter by letter.
Now, this copying isn't random. It follows some very strict rules, like a code. A C on the DNA always pairs with a G on the new copy. A G pairs with a C. Easy enough, but here's the one little twist you gotta remember. RNA doesn't use the letter T. So whenever the DNA has an A, the mRNA copy doesn't get a T, instead it gets a U, which stands for uracil. That U is kind of like the signature that tells you you're looking at RNA.
Okay, so we've got our photocopy. Our messenger RNA is made. The recipe is written down, but it's still inside the library. Now it's time for a little trip. And just like that, this brand new mRNA molecule, holding those precious instructions, zips right out of the nucleus. It travels into the cytoplasm, that big, bustling kitchen of the cell. Mission accomplished. The recipe has made it from the secure library out to where all the action is.
All right, the recipe has arrived in the kitchen. Now it's time to actually cook. This brings us to the second huge step in the process, and it's called translation. And that's exactly what we're doing. We're translating the language of the recipe into an actual physical dish.
So, the recipe's here. Great. But how does the kitchen crew read it? I mean, it's written in this language of A's, U's, C's, and G's. That language has to be translated into the language of proteins, which is all about amino acids. So how does the cell pull off this translation?
Here's the secret. The cell doesn't read the recipe one letter at a time. That would be crazy. Instead, it reads it in 3-letter words. These little words are called codons, and each 3-letter codon is the code for one specific ingredient, one particular amino acid. And those amino acids, remember, are the building blocks of our protein. Okay, so to do this, you need a kitchen crew. And we have 2 main players. First, you've got the head chef. This is a structure called a ribosome. The ribosome's job is to grab onto the mRNA recipe and read it. Then you have all the little kitchen assistants. These are called transfer RNA, or tRNA. Their job is to go grab the exact ingredient, the right amino acid, that the recipe calls for.
So you might be wondering, how does a little tRNA assistant know which ingredient to bring? Well, it's a brilliant matching game. The recipe, the mRNA, has its three-letter word, the codon. The assistant, the tRNA, has a matching three-letter anticodon on it, like a label. And when the label on the tRNA perfectly matches the word on the recipe, bam, it drops off its amino acid. It's a perfect fit every time.
And this whole assembly line happens with incredible speed and precision. It's broken down into 3 stages. First is initiation. That's when our chef, the ribosome, finds the start instruction on the recipe. Then comes elongation, and this is where the magic happens. The ribosome just chugs along the mRNA, reading word after word, while the tRNAs keep bringing in the right amino acids and linking them together into a chain. And finally, you get termination. The ribosome hits a stop word in the recipe, and the whole thing comes apart, releasing the brand new finished protein. So let's just take a step back. We've gone from a cookbook, locked in a library, to a finished meal ready to be served. We went from a secret code in our DNA to a real working protein out in the cell. Now let's talk about why this is so unbelievably important.
This two-step dance we just walked through, transcription and translation, this is it. This is how the information in your genes becomes you, how it becomes physical reality. It's also how your cells know what they're supposed to be. A muscle cell only cooks recipes for muscle proteins. A brain cell only cooks recipes for brain proteins. It's this selective reading of the cookbook that makes every cell in your body unique.
So yeah, to boil it all down, translation is simply the process where that genetic code gets decoded to build a specific chain of amino acids. And that sequence, that specific order of amino acids, is everything. It's what tells the protein exactly how to fold up into a complex 3D shape, and that shape determines exactly what job it's going to do.
This process, it's just so elegant and so incredibly precise. It's the absolute foundation of your health, of all life, really. Every single thing your body does depends on this working perfectly, trillions of times every second. Which, of course, leaves us with one last big question. If the whole system is this perfect, what happens when it's not? What happens when just a single letter in that master recipe gets messed up?